there's a big interaction between the cancer and the immune system. We'd like the immune system to do its job and make the cancer go away. But until that day, we will still need to learn more about the immune system, about the cancer, so that the immune system can actually win. There's a lot of genes involved in cancer pathogenesis, and Carol Prives has studied the most mutated gene in cancer for her whole career, which is P53. She was a pioneer in the field and has published seminal papers on the function of normal and mutant P53. When P53 started, it was an oncogene. That's before we knew the term tumor suppressor gene. And work by Carol and others in the field clarified that actually the mutant form had different functions than the wild type form, both in the neomorphic as well in the antimorphic fashion. And Carol has continued to pioneer many observations in this space at her professorship at Columbia. So today she's going to talk to us about how a particular pathway involved in metabolism is a regulator of cancer cell pathogenesis, and particularly that involving P53, which is the mevalonate pathway. Thank you.
Okay, thank you very much. It's a very nice introduction. Am I on? Am I mic'd up, as they say? It's great to be here. I have no problem with your tweeting anything that I've saying. Just don't put sad at the end of the tweet, okay? All right. The title that's in the program was just me not figuring out what I was going to talk about. So I've kind of focused in a little more, and I know what I'm going to talk about, which is the mevalonic pathway, which I kind of know how to pronounce usually. As Dave said, I've worked on wild type and mutant P53 for an embarrassingly long period of time, decades. And I've seen the progression of clarification of this protein from its first discovery when it was assumed to be an oncogene, and it sort of acted like an oncogene because, in fact, the versions of P53 that people first cloned were from cancer cells, and they were indeed not wild type, and they indeed did have oncogenic properties. And then it was discovered in the late 1980s by Bert Vogelstein and Arnie Levine and Moshe Oren that actually wild type P53 can suppress growth, and it fulfills the definition of a tumor suppressor. And so I'm going to talk to you about an aspect of P53 that we've been working on for a few years, and it actually deals, and I will be able to tell you that mutant forms of P53 can regulate this pathway in one way, and wild type P53 regulates this pathway in the exact opposite way. So P53 is a major tumor suppressor. I don't have to go through this in great detail. Highly frequently mutated in some cancers, less so in others. Occasionally, we believe that when you don't see mutant P53 in a tumor, it's because some aspect of the P53 network itself has been ablated, which is sometimes the case, for example, in melanomas. The reason why we know that P53 is a tumor suppressor is largely from mouse experiments. If you knock P53 out of a mouse, the mouse is a fully penetrant mutation. The mice will always get cancer, usually lymphomas and sarcomas. But even humans can tell us that P53 is a tumor suppressor. There is a class of families that have very high disposition to a set of cancers, not all of them, but several of them. And they have germline mutations in P53, and their cancers occur much earlier than the general population. I personally have been very involved with, I'm getting involved with studying cells from these patients. I think they're extremely interesting, and they actually could, in some very vague way, allow us to maybe learn how to treat these patients in a more effective way. And finally, the P53 was discovered as a protein binding to a viral protein, SV40 T antigen. Then several other DNA tumor viruses were shown to bind to P53. There is great interest in trying to harness our understanding about P53 to identify new kinds of molecules. There are molecules in the clinic that separate wild-type P53 from its main negative regulator, Mdm2, or MDMX is another one. And in some cases, there is a hint that in some settings, these drugs might be effective in certain cancers. Why is P53 so central? It's because it really sits at the node of multiple different pathways that themselves can be rationalized to be tumor suppressor, cell death, autophagy, cell cycle, and so on. And many of these, in fact, would be related to what we consider the good aspect of P53, the fact that it's tumor suppressive, modulates stem cells that even can be helpful in fertility. But too much of a good thing can be a bad thing. And for example, having a hyperactive P53 can actually be linked in some cases to neurodegenerative diseases, to premature aging, and other kinds of related syndromes. One of the more recent interests in P53 have dealt with its ability to regulate metabolism. And metabolism, of course, is a sort of a general topic. It's very, it refers to many different enzymatic processes in cells, frequently in the cytoplasm or in the mitochondria. And now we can definitely link what I'm going to tell you about P53 to a very specific aspect of metabolism, which is this pathway, and I'll tell you about that in a moment. But first, I'll just remind you that when P53 is mutated, its mutational spectrum is quite unique for a tumor suppressor in that a quite high proportion of mutant versions of P53 are actually present at very high levels in cells. And they are missense mutations. They are mutations of single amino acids. It's sort of an interesting way to look at this. I think I need another, this guy. A tumor suppressor can be defined as a gene that's mutated in many places, and those mutations cause loss of function. An oncogene, as you might well imagine, for example, the beautiful talk from Daphne, involves very often a single or very small number of mutations that occur with very high frequency at specific points. Well, P53 actually has the characteristics of both a tumor suppressor and an oncogene, because there is a set of what we call hotspot mutations that occur with very high frequency at very specific locations. We know that they abrogate the wild-type function of P53, which I forgot to tell you is to serve as a sequence-specific transactivator. But also, we know that at least some of these are actually oncogenic in their own right. And I'll tell you a little bit about that for the first part of this talk. So the first part of this talk is actually a somewhat old story. It was the result of a spectacular MD PhD student in the lab, Will Friedpastor, who after his residency is now doing a postdoc with Tyler Jackson. I think he's doing great work there too. In fact, he may be working on pancreatic cancer. And at the time he was in my lab, we had access to some breast cancer cell lines that were generated by Jill Barganetti, where you could knock down P53. And these cancer cell lines had only mutant P53 in them. And so we took a breast cancer cell line, MDA468, and showed that with full levels of P53, the vast majority of the clusters in 3D culture conditions, that's the, by now very well studied and very well often utilized protocol that was first pioneered by Mina Bissell, and then Joan Brugge made some beautiful observations, and many other people have worked with these breast cancer 3D cultures. And what had been shown by Mina is that virtually any breast cancer cell line that you put into this culture doesn't form normal, well-ordered structures that are acinar-like in their appearance, and usually consist of a polarized set of cells with a hollow lumen. In these cell lines, in this particular cell line, very few of these clusters you see in 3D conditions actually have this morphology. But when Will knocked down P53, now suddenly there was a very high proportion of the cells had this more normal appearance. This prompted him to collaborate with Arnie Levine and Scott Lowe, with whom we have a program project grant, to get some more information about this. And a gene expression array was done, and a pathway analysis was done. And to my surprise, the most significant pathway that was altered was the biosynthesis of sterols. In other words, the mevalonic acid pathway, in other words, the pathway by which cells synthesize cholesterol, as well as many other important metabolites, including the isoprenyl moieties that are attaching RAS and RAS-related proteins to membranes, proteins that play a role, even coenzyme Q that plays a role in the electron transport chain. Glycosylation is also dependent on this pathway functioning effectively as well. So this mevalonic pathway was a big surprise to me, and it was somewhat intimidating at the time because I never even thought about it. It's also the target of these very important drugs, statins, that prevent hypercholesterolemia, and additionally, different kinds of inhibitors that block either farnesylation or geranylation have been identified, and they're continually being studied as for potential use in the clinic. I know it's complicated. There have been some disappointing trial results many years ago, but I think there's still a lot of interest in working on targeting these pathways. So what is this pathway? It's a pathway that consists of, I think, at least 20 genes. And these are the names of the genes that encode the different enzymes that catalyze the formation of these metabolites. And they are coordinately regulated, really amazingly in my opinion, by a single transcription factor, SREBP2. This is a transcription factor that I'll tell you more about that a little later, but it basically coordinately regulates every single one of these genes by binding to a specific element in the promoter, and it's exquisitely regulated by the level of cholesterol in the cell. So it's also, as I said, this pathway is also the target of statins, and I'll get back to discussing statins in a little while. But statins themselves were of great interest to Will, because he thought that they might actually be able to be used for experiments he was thinking of. And indeed, when he put either these 2 breast cancer cell lines into 3D cultures, was able to show that these statins at, let's say, pharmacologically relevant concentrations had a significant impact on the growth and even the morphology of these cultures. And what was very satisfying to both of us was that when you took one of the metabolites, and I'm sorry, this is messed up over here, mevalonic acid pyrophosphate, and added it back to the culture where he had knocked down mutant P53, he lost the ability to see, or at least there was far fewer of these morphologically reverted structures in the 3D culture, meaning that at least one of the reasons why or how mutant P53 regulates the malignant morphology of these cultures is through this mevalonic acid pathway. And this is an experiment that was done with xenograft tumors, showing that statins indeed can impact their development. This is not a big shock. So how does this work? Well, I'm not going to go through all this now. It's sort of an old story, but basically Will showed that mutant P53 is somehow associated with SREBP2, and this association is helping to stimulate the ability of this transcription factor to activate expression of these genes, and thereby convert them from the normal phenotype to the 3D morphological phenotype, less mutant P53, more normal-looking structures in cells. We collaborated at the time with Annalisa Boris-Dale, who had several breast cancer gene expression data sets that she was able to analyze with us. This was actually just sort of at the birth, I think, of being able to really work with TCGA. And she showed that, with her, we showed that there was a cluster of breast cancers that had very high levels of many of the genes in the mevalonic pathway, and these were associated with those tumors that had the highest frequency of P53 mutation. And in fact, there was even a link with patient survival. Those patients that had the highest level of these enzymes also had worse survival than those that had lower levels of these enzymes. So this is where we were. We've gone on to do some studies trying to understand in a more general way how mutant P53 works. We have evidence that it associates with and facilitates in some way the activity of the SWI/SNF chromatin remodeling complex. And we're trying to bring together these two observations and determine how this chromatin remodeling complex is actually regulating the mevalonic acid pathway. But, I'm running out of time, sort of. I'd like to now tell you about some new, soon-to-be published data, which is basically goes to a very interesting fact about the reciprocal relationship between wild type and mutant P53. Very often genes that are activated by mutant P53 appear to be repressed by wild type P53 and vice versa. Since the mevalonic acid genes were being increased by mutant P53, the question was, does wild type P53 repress these genes? And if so, is this significant in terms of cancer? And the answer to both is yes. So the experiments were spearheaded by Seung-Won Moon in my lab, and it's been a fantastic collaboration with Scott Lowe's group, particularly a former student, Chun-Ho Huang, and a current postdoc, Shauna Houlihan. And between the three of them, I think they've put together a really nice story. So first of all, in cells, Seung-Won showed that if you turn on P53 with Nutlin, it's one of these drugs that activates P53 by preventing its binding to Mdm2, you get less expression of many of these genes. Only 3 are shown here. If you take a pair of isogenic cell lines, HCT, with P53 or from which P53 is deleted, you get a much greater repression when you turn on P53 in the wild type cells with virtually nothing or very little in the mutant cells. And we even did a metabolomics experiment in collaboration with a group in Denver. And they showed that several of these metabolites are significantly increased in cells that lack P53. And again, these are isogenic cells. These are SK-HEP1 cells from which we've deleted P53. Then we tried to generalize this because the idea is this true for only a couple of cell lines? It turns out that if you look at the cancer cell analysis, Cancer Cell Encyclopedia, in fact, you can get a very large number of cells, look at their gene expression, and it turns out that, in general, many of these mevalonic pathway enzymes genes are greatly underexpressed in tumors with wild-type P53 compared to tumors that either lack P53 or have mutant P53. What we then went on to try to figure out how this is working. So the first thing that Seung-Won did was to do a chromatin immunoprecipitation, looking now at the ability of this transcription factor, SREBP2, to bind to its element in one of the promoter for HMG-CoA reductase, significantly more binding in the absence of P53, which made us start thinking about SREBP2 as a target for P53. So let me tell you briefly what SREBP2 how it works. This is it's found in the ER as a large inactive precursor. Cells are exquisitely sensitive to the level of cholesterol, and the ER is a very big sensor of that through this complex between SREBP2 and SCAP. When cells are somewhat low in cholesterol, it can drop as little as 10%. This triggers a chaperoning of SREBP2 to the Golgi, where it's then subjected to 2 cleavages, releasing the mature transcription factor, which then gets into the nucleus, binds to an SR element, and so on. And so we thought maybe P53 is regulating this process, and indeed, we could show, and I can show this in many different cell lines, in the absence of P53, you always have more mature SREBP2 than in the presence of P53. A couple of other examples. In SK-HEP1 cells, activate P53, see less mature SREBP2, turn on P53 in another cell line, you again see much less of the mature form. So P53 is somehow regulating maturation. And now I'm going to tell you how this works. And if one could actually have a period of time that I show between slides that would somehow represent the amount of time it took to figure things out, I would have gone very fast for the first part, and then we'd sit around for 2 years waiting for the next slide. That's how long it took to figure out how this works. And it involved, first of all, painstakingly going through every single step that we know is present in the maturation and ruling out that as being a P53 target. And it also involved, and you heard Lou Cantley talk about reading the literature very carefully, because this is what young people should do. It involved Sung-Won carefully reading the literature, trying to figure out, trying to connect what he was seeing with how SREBP2 maturation has occurred. And it turned out that he figured it out. It turned out that a specific member of a class of ATP binding cassette transporters, ABCA1, is in fact the target that connects P53 to repressing mevalonic acid gene expression. So there are multiple ABCA1s. In fact, this one over here happens to be a pretty well-known P53 target, but it's this one, ABCA1, that turns out to be the critical target. How did he figure it out? By reading the literature, he came across a paper from a Japanese group who provided a very, I guess, paradoxical or unanticipated result. ABCA1 is most well understood as a protein that exports cholesterol, synthesized cholesterol, out of the cell to be dispersed elsewhere. What these groups showed was that it can also serve as a retrograde transporter of cholesterol from the membrane to the ER. They had very nice data for that. They even showed data on SREBP2 maturation. We then looked at our own RNA-seq data from a previous study and found that P53 upregulates ABCA1. And Joaquin Espinosa had done a beautiful experiment published a couple of years ago where he was looking at direct targets of P53. And among the very early direct targets is ABCA1. Also, ABCA1, among many different lipid transporters, is really significantly downgraded in tumors compared to the normal tissue. So it has those characteristics that it could be a tumor suppressor as well. So the next thing was to show that ABCA1 is a P53 target gene, and indeed it is. You induce P53 with nutlin and SK-HEP1 cells. You produce a lot of ABCA1 message and protein. In a cell line that we've engineered to have inducible P53, you induce a wild-type P53, you induce ABCA1. You induce a P53 that has mutations in its activation domain that prevent it from being a transcriptional activator, you don't get ABCA1. Furthermore, there are P53 binding sites in a couple of regions of the ABCA1 promoter, and we were able to show P53 associates via chromatin immunoprecipitation. So the question then is, if P53 is inducing ABCA1 and ABCA1 is repressing SREBP2, can ABCA1 do this without any P53 being present? And I think that's an important experiment because it really sort of focuses in the pathway on the relationship between P53 and ABCA1. So we have several cell lines from which we've deleted P53 by CRISPR. It's actually quite easy to do that. That's easy for me to say. I could probably never do it. But it can be done, and other people have done it as well. So we have cell lines, for example, SK-HEP1 with P53 knockout cells, and many other cell lines as well. And it turns out that at least some of these where you can actually find ABCA1, you can turn it on with an agonist, LXR agonist, which has this designation. And under those conditions, you definitely see less mature SREBP, less mature SREBP2. If you even knock down ABCA1 and then do the same experiment, you don't see this. So this is a link between ABCA1 and mature SREBP2. And so for the last part, and I'll go through this hopefully quite quickly, these are work that were done actually in Scott Lowe's lab that really validated these results in human cell lines in mouse models of hepatocellular carcinoma. Remember, cholesterol is largely synthesized in the liver, and so this pathway is very active there. Several years ago, Scott's group had published a paper where they had generated mice where the mice were expressing an shRNA directed against P53 that could be repressed. So the mice developed all the typical tumors that mice do, but then when they repressed the shRNA, the tumors actually shrank, indicating that once you turn on P53, you can now actually shrink a tumor in vivo. We got cell lines from them, and we could show that you could turn on P53 with these cell lines. And when you turned on P53, first of all, this is a volcano plot, virtually all to different extents of the mevalonic pathway genes are repressed, and ABCA1 is significantly increased. Furthermore, you could now do an experiment, which I'm going to describe in a little more detail in a second, where you can generate liver tumors in a very rapid way by injecting, using hydrodynamic tail vein injection, various constructs into the mouse, and those constructs end up being dispersed in the liver via the portal vein. I don't fully understand how this works, I've got to admit, but it does work. And these constructs would include MYC. It can also be done with RAS, mutant RAS, and CRISPR constructs that target either P53 or another tumor suppressor, axin, which is a component of the Wnt signaling pathway. And then you will get, in either case, within a few weeks, 4 to 6 weeks, lots of liver tumors in either case. But only in the P53 knockout tumors is there a huge increase in the amount of mature SREBP2, indicating that if you have wild-type P53, it's repressing it. So this led Chun Hao to do a very, I think, interesting experiment, which was to ask what happens if you don't let this mevalonic acid pathway get activated in these P53 null tumors. And to do that, he ablated P53, introduced MYC, and also a series of shRNAs targeting different components, different genes in the mevalonic acid pathway. And when that took place, there was a really striking reduction in the number of tumors. In other words, those tumors that could not make different components, HMG-CoA reductase, squalene, and lanosterol synthetase, and so on, they were much less. There were much fewer tumors, essentially, than in the control shRNA tumors. And this was not the case when the same experiment was done with axin. So then you got lots of tumors in those cases. So this was, I think, a stark example of loss of P53, not only increasing the levels of these enzymes, but actually creating a dependency on these enzymes for the tumor to progress. And so very recently, Shauna did a really beautiful experiment that I don't think anybody has done so far, is to ask, in the same protocol, what happens very, very early on, before you actually see a tumor? So instead of waiting 6 weeks, she essentially injected these into either wild-type mice or into P53 null mice, this construct, MYC, and then looked for the appearance of tumors, and then did an analysis of the genes using RNA-seq within 6 days. And these are actually cells that are just, we know we're expressing these constructs because they have GFP, but they're not yet tumor cells. And she showed that, in fact, even at this early stage, there's a big increase in mevalonic acid gene expression only in the absence of P53. This is just some more of these analyses over here, whereas ABCA1 was somewhat increased under these conditions. So I think that really implies that this is a very early stage in tumorigenesis, the creation, essentially, of very active cholesterol biosynthesis pathway if you don't have P53 there. So the more practical experiment, then, is to see whether you can actually pharmacologically intervene based on this knowledge, and in other words, treat the tumors with a statin. And indeed, when the P53 null mice were pre-treated with statins before the tumors were allowed to form, there was a significant reduction in both the weight of the tumors and the tumor numbers, as well as the weight of the organ as well. And this was not the case with the axon model. So that again, the statins were being effective only in the context of having no P53 present. So the last thing I'm going to show is something that I think kind of closes the circle. If P53 needs to activate ABCA1 in order to repress the mevalonic acid pathway, then what happens if you don't have ABCA1? In other words, is ABCA1 itself a tumor suppressor in this model? And it is. This is P53. This is just Mick alone under this time course, and this is ABCA1. This is actually a mixture. This is a composite of several different guide RNAs, some of which were much more dramatic, actually, than this particular sort of composite diagram over here. But what we could see here is that indeed ABCA1 is a tumor suppressor. You might ask, why is it not phenocopying P53? And so the answer is nothing will phenocopy P53. You can knock out any one of P53's extraordinary constellation of genes that are all perfectly well rationalized to be tumor suppressors, and you won't get this. But you'll be very lucky, actually, if you get this, as a matter of fact. So we're very pleased with ABCA1. And there is our data. And there is evidence in the literature from other people's work using different models that ABCA1 is a growth suppressor. In fact, isolating material from these tumors and growing cell lines out of them, what Sung-Wan has been able to show is that if they don't have ABCA1, they also have a much higher level of mature SREBP2. So that's the story. Putting together both our work on mutant P53, which we know can somehow activate via SREBP2, this pathway. And the somehow is a big word. We don't know how it works, to be honest. Hopefully we'll figure that out someday. Whereas wild-type P53, through activation of this target gene, shuts down this maturation process and allows, therefore, allows the cells actually to be protected from having runaway cholesterol biosynthesis. And when I say cholesterol biosynthesis, I don't know if it's cholesterol or isoprenoids or any combination thereof. It's a very important pathway in cell biology. So that's the story. This is my lab. Actually, I just realized I need an updated picture. I like to show this picture because of all the different countries that the people in my lab come from. And in fact, I bet that's true of everybody else as well. So science is a very international subject. The United States is still a magnet for people to come from all over the world. This is Sung-Wan, who did all the work on wild-type P53 repressing the mevalonic pathway. This guy over here, who's also an MD PhD student, made all sorts of CRISPR knockout and knock-in cell lines for us. And I really hope he comes back to join my lab. And I won't tell you about all these other people, but they're all doing great work. Thank you very much.
That's a great talk. So can you comment on what people's thoughts on how does sterol pathway promote tumor initiation in your case, and also the progression of tumor? Just sort of what's the general thought? Also linking back to the converting the 3D, the paper that you guys published, like what's really happening there in terms of the metabolism regulating the morphology? What's your thoughts on that?
So I think what you're asking, I think what you're asking is, in the context of the 3D cultures, if you knock down mutant P53 and you no longer get expression of these genes, how is this affecting the morphology? It's a great question. I don't know the answer to it. I really don't know. I mean, I don't know how you explain that functional reversion very easily, other than we feel we've strongly implicated that pathway in preventing that reversion from taking place. It's a good question. Daphne.
Evidence that statins are protective of liver cancer, and
Yes,
other quick question.
yes, there is. Okay, and I should have pointed that out. Actually, there's a lot of evidence that that patients that are on statins have a lower incidence of liver cancer. Whether there is any evidence that treating patients that already have liver cancer with statins is effective, that's much more questionable. And I should point out that the experiment we did with statins in the mice was the mice were given statins before we even started the tumor protocol. We have only anecdotally one experiment that was done when we let the tumors start to form and gave them statins, it was not effective. So it's more like a protective or a prevention measure than I'd say a treatment measure. I'll also say that a lot of these analyses involve, very large cohorts of patients, and liver cancer is very often involved with, there's a viral component of liver cancer, both HBV and HCV. And, the relationship there between P53 is not that well understood, I would say, at this point.
And do you need homozygous deletion of P53?
Excuse me?
Do you need homozygous deletion?
You mean in the human tumors?
In this particular model?
Yes, you need homozygous deletion, I believe. We never actually looked at hets. I believe that. Yeah. It goes to another question which we discuss a lot. It's very rare you have a human tumor that has both a wild type and a mutant allele. It's either one or the other.
To follow up the statin issues, and early on in early 70s and 80s, it was hard studies trying to correlate cholesterol levels and taking statins to heart diseases. The study found that indeed significant reduction in cholesterol correlate with increase in incidence of cancer, including colon and liver and other cancers. Can you make a comment on that, if a statin pathway could be as an anti-cancer drug?
First of all, I haven't looked at those studies, but I did look at in the last 5 years. I just looked through all the studies that have been done, and by now they're all meta-analyses, so they're combined studies where they do this. And by my count, 15 out of 17 concluded that statins have a beneficial effect for liver cancer, and I was only looking at liver cancer, okay? 2 of those studies said there was no effect. That's what there is. So it seems to me there's a preponderance of meta-analyses that would support the idea of statins being protective. Again, you don't know what the etiology of these patients are, whether there's a viral component or not. You just don't know. And I can't address other cancers.
Tend to be present at different amounts. In other words, there's a lot of mutant P53.
Yes, absolutely.
So is that influencing the biology? Does that mean just the amount? Does that change the target?
I think that's a great question. I mean, the experiment the problem is that if you bring wild-type P53 up to the same level as mutant P53, you're going to cause either cell cycle arrest or cell death. The mutant P53 is sitting there in the cell all the time with at least similar amounts of P53, if not more. And if you knock it down in certain cell lines, but not others, the cells will arrest even growing in 2D. They'll change their morphology in 3D. They'll invade less. I mean, there's a whole list of things that mutant P53 is purported to do. These are all cell biology experiments, and it's just a question of levels.